All right, this is Alzheimer's and its cell signaling, and it is commonly known for the dissociation of memories and cognitive thoughts, as seen here in this picture. So, what is Alzheimer's? It's a progressive disease that destroys memory and vital functions. It's the death of brain cells and connections, and there's no cure and cure. Alzheimer's is also called senile dementia, so senile dementia, and there are around 3 million cases in the US alone every year. And while Alzheimer's itself doesn't directly cause cancer, its destruction of the brain cells and the connections, and the destruction of mental function leads to the loss of motor functions. And when you lose your motor functions, like your ability to breathe and eat on your own, it becomes harder to live, and eventually you do die. So Alzheimer's isn't a direct cause for death in, immediately. It's a slow and arduous process. And now there's also a charity. There's the Alzheimer's Association, and they do a walk to raise money and awareness for Alzheimer's. Here is a scan of the brain. Here you have a healthy patient. Here's someone with minor cognitive damage, and here's someone with Alzheimer's. You can see the great disparity and difference. Uh, you can see the deterioration in the Alzheimer's brain while the healthy brain is fairly healthy, as the name implies. Now the one with minor damage, you can see small amount of damage here, not as vast as the Alzheimer's. I mean, you could probably pinpoint small amounts in the healthy brain, but those are normal. Now, important vocab to know is cleavage, and this is an important aspect of the brain and the cause of Alzheimer's. It is the division of a cell, the moderated removal or cut of a cell. Apoptosis, that's the cell-regulated death, and it's actually different in Alzheimer's. It's, I would say it's improper apoptosis because it's not directly called for by the body. A different signal is cueing it and bringing it to death. So it's not killing itself for the health of the body, it's actually causing damage. A ligand, it's a molecule that binds to a receptor in the reception stage. And what leads to this mutation or causes the signal to change is actually unknown in Alzheimer's. Um, then there's the extracellular amyloid plaques, and I'll go into further detail later, but these are the deposits of the amyloid beta protein, mainly in the gray matter of the brain, and this does lead to the destruction of the brain. Intracellular neurofibrillary tangles, these are abnormal accumulations of a tau protein that collects inside neurons. A tau protein is just a general name. It's a given name to a protein that's associated with Alzheimer's, and I'll explain later what it does. Um, acetylcholine Acetylcholine is a part of the brain that's associated with learning and memory, and so if any damage comes to this part of the brain, you will have severe uh, functional and motor function loss. Now, the intramembrane proteolysis, that's a widespread process by which cell transmits information from one compartment, specifically the endoplasmic reticulum, to another, the nucleus. And this is another aspect of Alzheimer's that is affected and that leads to the death. So, in the healthy state, you have your enzyme that's doing the cleaving, and cleavage is again the modular division of a cell to induce a reaction. You have your alpha secretase that's cleaving this. So it's cleaving your amyloid precursor protein, so your APP, your amyloid precursor protein, that's being cleaved. And in this diagram here, you can see that your APP is cleaved into an SAPP alpha, and a C83. Your C83 is a carboxy terminal fragment, and your SAPP alpha is actually associated with normal synaptic signaling. So this is your synaptic signaling, and this is healthy. So this is your healthy state here. So when you have an SAPP alpha and your C83, that's your healthy reaction in your healthy brain. Now in the disease state, where it actually goes wrong and your neurons begin to decay and they die, that's when instead of using this alpha secretase, you're actually using beta and gamma. And your beta can also be called BACE1 or memsapin 2. It's a protease that makes specific cuts during the maturation of some protein chains, so specific cuts. And your gamma actually catalyzes your intramembrane proteolysis of various types. Catalyzes intramembrane proteolysis. And if you remember, intramembrane proteolysis is the transmission of information through compartments from the endoplasmic reticulum to the nucleus. So it's increasing the speed. Now, what's actually happening is so instead of because you're not using your alpha secretase and you're using your beta and gamma, what you're getting a different when your cleavage is giving you a different outcome. So originally you had your APP, which was your alpha, which created your C83 and your SAPP. When it's cleaved with your beta and your gamma, you get something called A40 slash 42. So A40 and 42. These are harmful. These are neurotoxic fragments. So these are neurotoxic fragments, and they will aggregate. And so when they collect, you get a large amount of you get a plaque formation, and the plaque formation can cause serious damage to the brain. You get difficulty your ion channels get blocked. So your ion channel is blocked. This is your negative reactions. Your ion channel is blocked. Your calcium homeostasis is disrupted. So calcium homeostasis is disrupted. Your mitochondrial oxidation is stressed. So your mitochondria is harmed. Your energy metabolism is disrupted, and so is your glucose regulation. You have an abnormal glucose regulation. All of this will lead to your neuronal cell death, aka apoptosis. Now, again, as it's different, the signal itself is botched, and you're not supposed to have apoptosis, but it's killing the cell. These are leading to this. Your beta and your gamma are leading to the death of your cell. It's not proper, it's not intentional, and that's what causes Alzheimer's. Now, there's also another way that it's characterized, and that's with neurofibrillar tangles. So these tangles are the result of hyperphosphorylation of the microtubules associated protein tau. So hyperphosphorylation. And again, the phosphorylation is moderated and catalyzed by kinases. So you have your PKC, PKA, ERK2, and these are some of the ones that are involved in the hyperphosphorylation of tau. Here you have your tau. So this is your healthy, this is your diseased. You have your tau here, and this is your microtubule. Now, here you can see you have the formation, as I said, the neurofibrillary tangles. So these are your neurofibrillary tangles. Um, and you can see that these tangles and the hyperphosphorylation will lead to the disintegration of your microtubules. So these are disintegrating. And you can see it visually. In the healthy, they're a big, it's like a cone shape. It's fairly solid. This one you have the little particles, the molecules leaving and disintegrating from your neuron. So here again, we have another example. Here's your normal brain or your Alzheimer's brain. Here you have a regular neuron. As you can see, the surface of it is fairly flush and it's smooth. Here, however, you see the neuron surface is fairly granulated and it's unhealthy. It can be it's there's a visual difference. Um, and here you can so here you have your neurofibrillary tangle. So here inside of the neuron is your tangles, and then here is you have your amyloid plaque buildup. So again, your plaque buildup is going to cause a lot of damage to the cell, and it's going to lead to the neuronal cell death. Now, the your what should be known is that your neurofibrillary tangles are actually caused by the tau undergoing oligomerization. Z. And that leads to the apoptosis. So this involves the contracting of an olefin with a catalyst in order to produce a larger chain molecule. And there's a lot of things going on. The hyperphosphorylation and the increased chain is leading to the destabilization of your neuron, leading to disappearance here and here, and these plaque buildup. Again, all of this, your neuron, your neurofibrillary tangles, and your plaque buildup, and your microtubule disintegration will all lead to your neuronal cell death or your unintentional slash improper apoptosis. Now, this is important, again, because the root cause is not known. We don't know what's causing this, what's signaling this reaction, what ligand is causing it. That's why there's no cure, because we don't have a true understanding. We're just now learning the proteins that are involved in the cleaving and the signal transduction that leads to the apoptosis being activated, but we don't know what's actually telling the cell to undergo this reaction. So there are these charity events to help. And I encourage you to do your own research because it is a very fascinating disease. It's very tragic in the way that it does dissociate memories. Um, yeah, and that's Alzheimer's.